Thank you for the introduction, and I'm glad to have this opportunity to be here to talk about the latest progress in NGS-based biomarkers, and the main focus is on precision immunotherapy. Of course, we all know that, this is my disclaimer. Of course, we all know that the way that immunotherapy for oncology works is because during the tumor progression, you also have tumor cell death, and the release of tumor-associated or tumor-specific antigen was released into the environment, which stimulate our immune response. And later, this immune response can elicit immune surveillance. And when the tumor begins to grow, this normal tumor suppression or immune surveillance was suppressed by a lot of checkpoint molecule. And so most of the already approved checkpoint inhibitor worked by blocking the checkpoint molecules. So the first cycle, or the first molecule that's kick off the cancer immunity cycle, is the release of tumor antigen. But we also know that through the past few years, we already witnessed the efficacy of a lot of checkpoint inhibitor. And for most of the cancer type, the drug is effective, except it's only effective for a small portion of patients. So in that case, we definitely need to have some biomarker in order to identify those patients. And since most of the checkpoint inhibitor either work on PD-1 or PD-L1, so original idea is to develop or use immunohistochemistry to identify those tumors which express high level of PD-L1. And that's why most of the checkpoint drugs, when they enter into clinical study, they're evaluated for their patients' PD-L1 expression level. And for some drugs and for some cancer types, that strategy works quite well, but it doesn't work for most of the cancer type. So the question is that whether we can also identify another biomarker. And so through the documentation of the response rate of those checkpoint inhibitor blockade, we also observed that for those tumors which have higher tumor mutation burden, they tend to respond better to PD-1 or PD-L1 blockade. And so this fits quite perfectly with the concept of that when the tumor has more tumor antigen or more mutation, it tends to have more chance to elicit immune response. And if you can take away the suppressor, then the immune system can work to fight the cancer. And so tumor mutation burden has become a marker, or at least marker that have been in the high hope to work as a biomarker to select those patients who likely would respond to checkpoint inhibitor. And as you can see, this is also the evidence that for those tumor type that has higher tumor mutation burden, their response rate to PD-1, PD-L1 treatment is much higher. So tumor mutation burden is a measure of the number of somatic mutation in the tumor. But typically, tumor mutation burden is monitored or measured by using whole exome sequencing, and then we count how many mutations are present in the patient's tumor. It works quite well for research project, but if we're going to move this into clinical setting, conducting whole exome sequencing for every patient is virtually impossible. And the main reason is because for whole exome sequence, you do need to have high quality tumor sample plus matched normal. And those tumor sample has to have sufficient tumor content. So it's not possible. Therefore, and also it's much more expensive in the clinical setting. So everyone thinking maybe there's a possibility that we can use, instead of whole exome sequencing, we can use a panel sequencing to also derive tumor mutation burden. And actually the speculation that tumor mutation burden can predict the efficacy of treatment response have already clearly been illustrated in the CheckMate 227 result. So for this particular trial, they have already identified that for any patient who has more than 10 mutations per megabase, the patient will have much better response in terms of response rate, in terms of the overall survival, as well as the duration of survival. And so FDA is looking into approving TMB as a biomarker for immunotherapy. There's only one problem. If we are not going to use whole exome sequence, when we use panel-derived sequencing technology to represent tumor mutation burden, what kind of panel sequencing we need to have? And would the panel sequencing actually reflect the tumor mutation burden identified from whole exome sequencing? So this is a publication published early last year that shows that if you use the whole exome sequencing compared to the result that you obtain from whole exome sequencing, you can actually see a pretty good correlation. So whole exome sequencing versus panel sequencing. And also, between two panel sequencing, you can get pretty good correlation. So this actually gives us some idea about how to use panel sequencing to reflect tumor mutation burden. But when we talked about whole exome sequencing, we are talking about sequencing 18,000 genes. But most of the whole exome panel sequencing, we're talking about a few tens of genes or hundreds of genes. So how many chromosomal regions you need to cover in order to get a decent result to predict tumor mutation burden? And based on the study presented by Foundation Medicine, if you want to determine the tumor mutation burden of about 10 mutations per megabase, you need to cover at least 1 megabase of genome. So these are all studies done by a single company. But in addition to Foundation Medicine, there are also many other panels that have been approved or on the process of being approved. So for example, Memorial Sloan Kettering has a panel that covers 468 genes. And Genomics has a panel that covers 440 genes. And we all claim that we can measure or calculate tumor mutation burden. And in addition to our panels and the other two panels, there are hundreds of companies claiming that they can calculate tumor mutation burden. So this creates a big problem. Most of the panel sequence, when they finish the sequencing, they will present tumor mutation burden. And this is going to be the data that will be used as a biomarker in the clinical setting to select patients who likely respond to checkpoint inhibitor. But when there are 10 or 20 companies, they all use different panels, different size of panels, different method, different algorithm to calculate tumor mutation burden. Which tumor mutation burden predicted by different panel is more accurate, or at least more closely resemble to the whole exome sequencing tumor mutation burden? And if FDA approved TMB as a biomarker, should they also try to standardize the method or at least harmonize the method so that when different companies talked about tumor mutation burden, at least physicians or drug companies have idea how well this different TMB, panel-derived TMB compare to each other and compare to whole exome sequence. So in order to prevent the later confusion in the clinical setting, the TMB harmonization program was kicked off by Friends of Cancer Research Foundation in the US, and it's actually backed by US FDA and NCI. So the whole purpose of the TMB harmonization program is to compare the TMB derived from different panels and compare their performance to whole exome sequencing, and trying to go through this harmonization program to facilitate how we quantify TMB and facilitate in the future to bring this marker into clinical setting. So the TMB harmonization program actually consists of 3 different phases. The first phase is that all of the diagnostic party will actually compare their in silico algorithm to see when you use a limited number of gene or limited genomic coverage and using your own computation algorithm, would you be able to faithfully or accurately predict the tumor mutation burden? And so this particular phase 1 study was kicked off in May this year and finished the initial study and data analysis just a couple of weeks ago. And this data was released yesterday, so about 12 hours ago in Washington, D.C. In the city meeting as a late-breaking oral presentation. And so luckily, because of that, data has already been released, so we can show some of the data and then also share some of our idea. So there are many different parties participating in this TMB harmonization program. Of course, you have government agency, both NCI and US FDA. You also have some academic institute, so Columbia University, Johns Hopkins, and Memorial Sloan Kettering. And there are 10 different diagnostic companies, so including Genomics, Carriage Life, of course, Foundation Medicine, Guardant Health, Illumina, NeoGenomics, OmniSeq, Personal Genome Diagnostic, Cryogen, and Thermo Fisher. So all of the big name companies involved in molecular diagnosis and next generation sequencing based technology are involved in this comparison. And then there are also a lot of drug companies looking into this particular program. So all of this data actually are compared. So the first step is to obtain data from TCGA pan-cancer study. So that includes about 4,000 samples with whole exome sequence data, cover 32 cancer types. We use 2 different approaches. The first approach is that we use a standard measure to calculate tumor mutation burden on the whole exome data. And then this data, the initial data set was also distributed into all of the diagnostic company. And so each company used their own panel and their algorithm to predict their panel-derived tumor mutation burden. And both data were sent back to NCI. So NCI will use this data and do a blind comparison. So all of the data that I show here is blinded. So we don't really know which panel represent which company's diagnostic method, except they do notify us that our panel is the number 2, panel 2 in this comparison. So the first thing that we compare is that how well the panel-derived TMB compared to whole exome sequence. So these are the data from all 11 labs. So here is the panel-derived TMB and whole exome-derived TMB. These are all similar or completely exact same data set. So you can see that most of the whole exome sequence data actually correlate quite well with panel-derived TMB. So this is a 45-degree line. A panel that performs well shall have a slope very close to the 45-degree line. And also, you can see the correlation that, and also the association. So most of the panel actually has a correlation with whole exome sequencing with the R square between 0.85 to 0.93. And as I mentioned, we are lab number 2. So this is our data that we have an Pearson correlation of 0.96 and an R-square of 0.92. So how well does this compare to other companies' panel? So this is the summary of those Pearson correlation as well as R-square data. So 0.96 is among the top performer that you have a very good correlation or linear correlation between tumor panel TMB and horizontal TMB. And so this just look at the correlation. But the next thing that we need to look at is because at this moment we still don't know what will be the best cutoff for the TMB high. So it could be somewhere between a mutation per megabase or 10 mutation per megabase or perhaps even 20 mutation per megabase. So we also, NCI also look at individual panels' performance at different cutoff for positive prediction positive prediction agreement as well as negative prediction agreement. So basically we are looking at the concordance between panel-derived TMB and horizon-derived TMB. So you can see that some companies' panel performs better at low low cutoff, while other companies performs better at high cutoff. And this is the panel 2. We actually have the similar concordance across different cutoff for both positive prediction and for negative prediction. So it doesn't matter which cutoff eventually will be used for TMB. At least our panel actually performed quite equally well across different cutoff. But this is looking at the concordance. The most important thing is that what about the precision and the accuracy? Okay. We also look at the horizon TMB and panel TMB and whether it actually work equally well across different cancer type. And it seems like panel TMB tend to overestimate TMB in certain cancer type, and it's similar phenomenon for different cancer, for different panel. But it actually worked quite well for most of those cancer that has full TMB range. But when we talked about precision, we're talking about when you use a panel TMB to predict horizontal TMB, when you have a 10 mutation per megabase prediction, is it very, very tightly associated, or actually the prediction is quite scattered? So here NCI actually did a very stringent test, and because we know that the most likely cutoff for TMB high will be at 10 mutation per megabase. So they took out the data in that data set and isolate all of those patients who has TMB somewhere between 0.5 and 10.49. So these are the patient group that we consider have 10 mutation per megabase, and then look at the performance of individual panel. And so for a panel that has high accuracy and high precision, all of the predictions should be very close to this 10 mutation per megabase baseline. And if the panel tend to overestimate, then you will see that situation like this. And so this is a very, very tough and real test about the algorithm and about the panel performance. And again, here is the panel 2. And so among all of this panel that's being compared, we actually has the tightest prediction for this data set. So that means that when we talked about 10 mutation per megabase using different panel, for this particular panel, you gotta wonder whether it's actually 10 mutation per mega or it's 3 mutation per mega. But for our panel, it's actually quite close to the real number. So based on this data that FOCR and TMB make the conclusion during the meeting. And so it seems that based on the TCGA data, the panel sequencing TMB actually correlate quite well with whole exome sequencing TMB. So it can be used in the future as a biomarker in the clinical setting. But different panels do perform differently. It may have something to do with the gene number, with the gene selected, or the algorithm used by individual panel. It seems to work quite well across different cancer types. But is this the last thing that we need, or is this just the very beginning? We actually know that in addition to use high tumor mutation burden to predict patient's response, there's also things that we can get from the NGS data, because you can use the panel sequencing data to accurately identify MSI status. You can also use the sequencing data to identify patients' genomic status in those immune-related genes. And this actually will help us to identify also those patients who has PD-L1 amplification, who are likely to respond to checkpoint inhibitor, or who has P10 deletion, who tend to be resistant. And then also there are a lot of genes or pathways, when mutated, they tend to convert resistance. So this information needs to be provided as well, instead of just counting the number of mutations. And finally, I think there's one thing that's really important and need to be taken into consideration very seriously. That is hyperprogression of the patient upon the treatment of checkpoint inhibitor. And previous data have already shown that hyperprogression occur in about 20% of patients in lung cancer and also in head and neck cancer. And the initial study identified MDM4, MDM2 amplification or EGFR mutation as a predictive marker for patients who tend to exhibit hyperprogression upon checkpoint inhibitor treatment. And I think for the future biomarker for checkpoint inhibitor, particularly using NGS technology, we need to provide all of this information in order to help patients to better understand their status and help physicians to determine who is better candidate and who should be should not be treated with checkpoint inhibitor, so we can realize personalized immunotherapy. Thanks.